Somewhere under the Antarctic ice, a fish is resting on black mud in water, cold enough to freeze its blood solid. It isn't frozen, cut it, and what runs out isn't red. It's almost clear. Every vertebrate on Earth carries hemoglobin. This one doesn't. It should be dead twice over, suffocating and freezing at the same time. The easy answer is that it barely moves at all, and that answer is wrong. Its heart is enormous. The antifreeze in its body isn't even the strangest part. The strangest part is the ice already inside it, ice that refuses to melt. To understand why this animal should not be alive, forget the fish for a moment and look at the water. Drop a camera through a hole in the sea ice off the Antarctic coast, and the first thing that hits you is not the cold. It's the clarity. The water is so clean and so free of the plankton bloom that clouds temper its seas for most of the year that the beam of light seems to travel forever before it fades. You can see the underside of the ice above you like a ceiling of frosted glass. You can see structures hanging from it. And down below, past the light, there is a floor of gray-brown sediment scattered with sponges and sea stars, and the occasional pale shape lying motionless, waiting. Now look at the temperature reading on the instrument package. In the coldest parts of this system, the water sits at about minus 1.9 degrees Celsius, not near zero, below it. The water is liquid at a temperature where fresh water would already be a solid block. That's not strange in itself. Seawater is salty, and salt gets in the way of ice formation. Water molecules trying to lock into a crystal lattice have to shove dissolved ions out of the way first, and that costs energy, so the whole process stalls until the temperature drops further. The saltier the water, the lower it has to go. Full-strength seawater freezes at roughly minus 1.9. So this ocean is not doing anything mysterious. It is sitting exactly at its own freezing point, which is where the coldest shelf water in the world tends to live, in constant contact with ice above it and ice frozen to the seabed beneath it. Here's the problem. A fish is not seawater. The body fluids of a fish are salty, but nowhere near as salty as the ocean. That's not an accident or a design flaw. Marine fishes with backbones generally keep their internal salt concentration at roughly 1/3 to 1/2 of the surrounding sea, which means they spend their lives fighting a slow osmotic leak, constantly losing water to the environment and constantly replacing it. The upside is that their cells work properly. The downside, in this particular ocean, is fatal, because a body fluid that is only 1/3 as salty as seawater freezes at a much higher temperature. In most fishes, that point lands somewhere around minus 0.7 to minus 1 degree Celsius, which means an ordinary fish dropped into water at minus 1.9 is not merely uncomfortable. It is roughly a full degree below the temperature at which its own blood is supposed to turn to ice. Sit with that for a second, because it is not a small margin, and it is not a seasonal problem. It is the permanent, everyday condition of this entire ocean. The water is colder than the freezing point of the animals living in it. Not sometimes. Always. So why isn't every fish on that seafloor a frozen statue? The first answer that occurs to most people is supercooling, and it's a reasonable guess, because supercooling is real. Pure liquid water can be chilled well below zero and stay liquid if nothing disturbs it. It needs something to start from: a nucleus, a speck of dust, a scratch on the container wall, a seed crystal. Without a starting point, the molecules can't organize, and the liquid hangs there in a fragile state, waiting. Fish blood can supercool the same way. In deep water, far from the surface and far from any ice, some polar fishes do exactly that and get away with it. And in shallow Antarctic water, that strategy is suicide, because this environment is not short of nuclei. It is saturated with them. Watch the underside of the sea ice through a diver's camera, and you'll see something that looks like a chandelier collapsing in slow motion. Thin, flat, translucent plates of ice, some the size of a hand, growing downward and sideways from the ceiling in loose crystalline mats. That's platelet ice, and in some places it forms a layer meters thick beneath the solid sea ice, a shifting mesh that fish swim through the way birds move through a hedge. Farther down, where supercooled water drains off the ice shelves and drifts across the shallows, ice grows directly onto the seafloor, encasing rocks and sponges and anything slow enough to be caught. Anchor ice. It builds until it's buoyant enough to tear free. Then it rips a piece of the seabed loose and carries it upward. Now put a fish in the middle of that. The threat isn't the temperature by itself. The threat is contact. A single ice crystal touching an exposed surface, a gill filament, the lining of the mouth, a scrape on the skin, the wall of the gut after the animal drinks seawater is all it takes. That crystal is a template. Water molecules in the fish's body, fluids, already colder than their own freezing point and desperate to organize, latch onto the lattice that's being offered to them and lock into place. Then more do. The crystal doesn't stay small. It grows into the tissue, spreading along the path of least resistance. And it does not need long. That is the actual mechanism of death here. Not slow chilling. Not the body shutting down degree by degree. A phase change, seeded by touch, propagating outward from the point of contact until the animal is structurally destroyed from the inside by its own water. This is the reason ice is more dangerous in shallow water than deep. The deeper you go in the Southern Ocean, the less likely you are to meet a floating crystal, and the more you can rely on supercooling to carry you. Come up onto the shelf, into the shallows where the platelet layer hangs and the anchor ice creeps. And supercooling stops being a strategy and becomes a countdown. Every one of those crystals is a loaded trigger, and the fish is swimming through a room full of them. So the animal on the seafloor has an exact, brutal, non-negotiable problem to solve. Not tolerate cold, not slow down and conserve energy. It has to swim, feed, breathe, and drink inside a fluid that is trying to nucleate it. And it has to do that continuously for a lifetime that can run into decades. It cannot solve this by raising its internal salt concentration to match the sea, because at that concentration, its enzymes and its nerves stop working properly. It cannot solve it by generating heat, because it's a fish in freezing water with a gill surface designed for gas exchange, and any warmth it produced would be stripped away almost instantly through those thin, blood-rich membranes. It cannot avoid ice, because ice is in the water column, on the ceiling, and on the floor. It cannot stop drinking, because it's a marine fish, and it drinks seawater to replace what it loses osmotically, which means it is deliberately swallowing the very medium that carries the crystals. Every obvious escape route is closed, and yet the seafloor down there is not empty. It is populated, not sparsely either. Across the Antarctic continental shelf, one group of fishes has taken over so completely that almost every fish down there is a member of it. This is not a place where life clings on at the margins. It's a place where one lineage of vertebrates found the loophole and then inherited an entire ocean. So there's something in these fish that stops the crystal. For a long time, nobody knew what it was, and the search for it started with a fairly simple observation that didn't add up. If you took blood or body fluid from an Antarctic fish and measured the temperature at which it melted, you got a value in the normal range for a marine fish. Slightly depressed, maybe, but nothing dramatic. Nothing that could explain surviving at minus 1.9. Then you cooled the same sample down and watched for the temperature at which it froze, and it didn't freeze where it should have. There was a gap. The sample melted at one temperature and refroze at a distinctly lower one, And those two numbers are supposed to be the same number. For a simple solution, the melting point and the freezing point sit on top of each other. That's basic physical chemistry. Salt water melts and freezes at the same temperature. Sugar water melts and freezes at the same temperature. There is no gap. Fish blood from the Southern Ocean has a gap. And that gap is the loophole. Whatever was in there was not doing what antifreeze is supposed to do. Think about how antifreeze works in a car radiator. You pour in a chemical, usually a glycol, and you pour in a lot of it. It works by brute force. Every dissolved molecule you add gets in the way of water molecules trying to organize into ice. And the more of them you add, the further you push the freezing point down. It's a numbers game. The effect depends almost entirely on how many particles are floating around in the solution, not on what those particles actually are. Salt does it, sugar does it, alcohol does it. This is the colligative route, and it has one non-negotiable cost. To move the freezing point a meaningful distance, you need concentrations that are enormous. That's the part that kills the idea for a living animal. To depress a fish's body fluids by even 1 degree using dissolved salts or small molecules, you would have to load the blood so heavily that the osmotic balance of every cell in the body would be wrecked. Water would be pulled out of tissues. Proteins would misfold. The kidney would be fighting a losing war. You would solve freezing by causing something closer to poisoning. The molecules in Antarctic fish blood are not doing that. They are present at concentrations that, measured colligatively, should barely register. They contribute a fraction of a degree by the numbers game, and that fraction is nowhere near enough to explain survival at minus 1.9. And yet the fluid refuses to freeze until you push it well past where it should have solidified. The effect is far larger than the concentration justifies, which means the mechanism is not about crowding the water at all. It's about the ice. These molecules are antifreeze glycoproteins, and their structure is almost absurdly plain. A repeating backbone of 3 amino acids over and over with a sugar group hanging off at regular intervals. No elaborate folded architecture, no catalytic pocket. Nothing that looks like the intricate machinery you expect from a protein doing something clever. It looks like a length of chain with beads on it. And it works because one face of that chain matches the geometry of an ice crystal surface closely enough to stick to it. That's the whole trick. It sticks. When a growing ice crystal appears in the fish's fluids, these molecules find it and adsorb onto its surface, pinning themselves along the planes where the crystal would normally add new layers. The crystal now has a problem. Ice grows by water molecules joining at the edges of flat advancing steps. Cover those steps with foreign bodies, and the water molecules can only add to the small gaps left between them. So the ice does keep growing, but it can only bulge outward in tiny curved bumps between the anchored proteins, and curvature is expensive. A sharply curved ice surface is thermodynamically unfavorable compared with a flat one. The tighter the bulge, the harder it becomes for the next water molecule to attach, and the more you have to lower the temperature to force it. So the crystal advances a little, curves, and then stalls, held in check by nothing more than geometry. To make it grow again, you have to chill the whole system further. That's the gap. That's why melting and freezing come apart. The proteins have not changed the temperature at which ice melts, because they don't interfere with melting at all. They have changed the temperature at which ice can grow. It is not a chemical shield, it is a physical handcuff. And the efficiency of it is absurd, because a tiny amount of protein disables a process that would otherwise be unstoppable. The margin it buys is not large in absolute terms. The freezing point of the fluids gets pushed down to somewhere in the region of minus 2 degrees, a few tenths below the seawater around it. That sounds trivial. It isn't. In this ocean, the water can't get colder than its own freezing point, so a few tenths of a degree of headroom is all the animal will ever need. The proteins are not over-engineered. They are tuned exactly to the environment, with a small safety margin, and not one bit more, which is what selection tends to produce when the pressure is relentless and the resource is expensive. Where the proteins sit is just as revealing as what they do. They are distributed through the blood and through the fluids that bathe the tissues, and they are concentrated in the places where ice is most likely to enter, not the deep interior. The border, the gut fluid, because a marine fish drinks seawater and swallows crystals with it. The tissues near the gills and the skin, because those are thin and in direct contact with the outside. A few compartments are left out: the fluid inside the eye, the fluid of the inner ear, the urine in the bladder, and those fluids sit undercooled, protected only because everything between them and the ocean is loaded with antifreeze. The defense is a perimeter, not a bunker. And this is where the story stops being a neat piece of biochemistry and becomes something stranger, because of where the protein came from. Sequencing work on these fishes traced the antifreeze glycoprotein gene back to an unexpected ancestor, a gene for trypsinogen, the inactive precursor of a digestive enzyme that a pancreas secretes to break down protein in food. Nothing about a digestive enzyme suggests ice. The connection shows up in the leftovers. The antifreeze gene kept the front end of the old digestive gene, the short signal sequence that tells a cell to export the finished product outside itself. And it kept a piece of the tail end as well. What sat between those two pieces was replaced. The middle of the old enzyme was, in effect, gutted, and in its place a tiny 9-letter stretch of code specifying 3 amino acids was copied over and over until it formed a long repeat. Read that again, because it is about as direct a picture as biology gets of how a genuinely new function appears in a lineage. A gene for eating became a gene for not freezing, not by inventing something from nothing, but by keeping the packaging, throwing out the contents, and duplicating one small fragment dozens of times until the repetition itself became useful. The timing estimates place that transformation within the last several million to roughly 15 million years, and those estimates carry real uncertainty because molecular dating depends on assumptions about mutation rates that are not fixed in stone. What matters is the rough coincidence. The gene appears to have been built during the same broad window in which the Southern Ocean was going cold and ice was becoming a permanent feature of the shelf. The tool and the crisis arrived together. Which of them arrived first, and by how much, is a question the evidence has not fully settled. There's a second detail that makes the whole thing harder to shrug off. Far away, in the Arctic, in a completely different group of fishes, there is another antifreeze glycoprotein. Same repeating 3 amino acid backbone. Same sugars. Functionally interchangeable in the lab. And it did not come from a trypsinogen gene. The two lineages have been separated for far too long in oceans that went cold at different times for this to be a shared inheritance. Two different groups of fish facing the same physical problem arrived at nearly the same molecule from different genetic starting points. That tells you something uncomfortable about how narrow the solution space is. There are not many ways to stop an ice crystal with a protein. Evolution found essentially the same one twice because the geometry of ice does not negotiate. So the freezing problem is solved. The animal can sit on the seafloor at -1.9 with crystals drifting past its gills, and it will not be nucleated. The handcuffs hold. And that solution creates a problem that is arguably worse than the one it fixed. Look again at what the proteins actually do. They do not dissolve ice. They do not destroy it, dismantle it, or push it out of the body. They grab it and hold it in place. The crystal is still there. It is still ice. It has simply been rendered incapable of growing. So what happens to it? The obvious assumption is that it melts. Antarctic water is not uniformly frozen year-round. In summer, in shallow coastal areas, sunlight reaches through leads and thin ice. Meltwater comes off the coast, and the water column warms. Not much by the standards of anywhere else on Earth. a fraction of a degree, sometimes a degree or so in the shallowest, sunniest corners. But that is enough to push the temperature above the melting point of ice. And once you're above the melting point, ice is supposed to disappear? Melting is not like freezing. It doesn't need a seed. It doesn't need a nucleus. Warm a crystal past its melting point, and it simply loses its structure, molecule by molecule, from the surface inward. There is no barrier to it. That's why ice cubes are reliable and supercooled water is not, which means the ice crystals inside these fish should be a seasonal inconvenience. Winter loads them up. Summer clears them out. The animal resets and does it again. That's not what the fish show. When researchers looked inside notothenioids caught during the warmer part of the year, in water that had already risen above the equilibrium melting point, they were still finding ice crystals in the tissues, not residual slush in an animal pulled from freezing water, ice inside fish that had been swimming for weeks in conditions where it should already have vanished. The explanation turns out to be the same mechanism that saved them, running in reverse. The antifreeze proteins are bound to the crystal surfaces. They are physically in the way. A melting crystal has to lose water molecules from its surface, and a surface that is coated in adsorbed protein cannot shed them freely. The protein stabilizes the thing it captured. So the ice sits there at a temperature where it has every thermodynamic reason to be liquid, and it stays solid anyway, held together by the molecules that were supposed to be protecting the animal from it. Ice that is warmer than its own melting point and still ice, superheated ice, inside a living vertebrate. That's an unusual claim, so it's worth being precise about how far the evidence goes. The stabilizing effect has been shown in controlled conditions, where ice held by these proteins can be warmed past the point where unprotected ice of the same size would have dissolved. Crystals have been recovered from wild fish in summer water, and long-term temperature records from one of the coldest coastal sites in Antarctica, logged year-round over more than a decade, show that the water there never spent enough time warm enough to overcome the effect and flush the ice out entirely. What is not settled is what that means over a lifetime. The fish here are not short-lived. Many Antarctic notothenioids grow slowly, mature late, and can live for a decade or more, some considerably longer. If each winter delivers a fresh load of crystals through the gills and the gut, and each summer fails to fully clear them, then the arithmetic runs one direction only. The ice accumulates, not enough to freeze the animal, because the proteins prevent growth, but it stays. Where it ends up is one of the more suggestive details. Crystals show up concentrated in the spleen, which is exactly the organ you would expect if the body is treating them as foreign particles. The spleen filters the blood. It pulls debris and worn-out cells out of circulation and holds them. If ice crystals are being swept up and parked there, that looks less like a leak and more like a containment strategy. Don't try to destroy the thing you can't destroy. Just move it somewhere it can't do damage. That's an inference from where the crystals are found, not a demonstrated process. And it's worth flagging as such. Nobody has watched a crystal make that journey. What the anatomy suggests and what actually happens are different categories of claim, and this sits in the first one. The long-term consequence is genuinely open. Does a fish that has lived 20 years in this system carry a measurably larger burden of internal ice than a young one? Does that burden eventually interfere with the spleen's other jobs? Is there a limit, a point at which the accumulation becomes a cost that shortens life? Nobody has run that experiment, and running it would be difficult, because it would require following individual wild fish through their entire lifespans in an ocean that is expensive and dangerous to work in for a few weeks a year. So what you're left with is a survival solution with an unpaid invoice attached. The animal does not defeat the ice. It arrests it, files it away, and carries it. Every year, it wins the same fight again. and takes on a little more cargo. It's not victory. It's an indefinite postponement. And the postponement is permanent because the environment never lets up. That pattern is going to repeat. Hold on to it, because it is the shape of this entire animal. Now, if you were designing a body to carry that kind of load, you would want it robust, redundant systems, backups, margin everywhere. Something that can absorb a hit and keep functioning, because the environment is relentless and there is no season when the pressure comes off. The fish at the center of this story went the opposite direction. It threw things away. Pull one of them out of a trap and lay it on the deck, and the first thing you notice is that it looks wrong in a way that's hard to name. The body is long, tapered, front-heavy. The head is broad and flattened, with a wide mouth full of small teeth, and the snout is stretched into something distinctly reptilian, which is why the family picked up the name crocodile icefish. The skin has no scales anywhere. It's bare, smooth, and slightly translucent, and through it you can see the shapes of things you are not used to seeing through skin. The pectoral fins are large and set low like paddles. The eyes are big and dark and dominate the front of the skull, and the gills are the wrong color. A fish gill should be red, deep red, sometimes almost purple, because it is one of the most densely vascularized tissues in the animal kingdom, packed with blood built to carry oxygen. Lift the operculum on an icefish, and the filaments are pale, off-white, faintly grayish or cream, like something that's been left in bleach. Cut into it, and the fluid that comes out has almost no color at all. Slightly milky, sometimes with a faint yellowish tint. It is unmistakably blood, in the sense that it clots, and it carries cells, and it circulates. It just isn't red. The first published account of this in the middle of the 20th century described exactly what anyone would describe: a vertebrate whose blood contained no significant respiratory pigment and effectively no red cells, which, for a vertebrate, is not an eccentricity. It is a category error. Hemoglobin is not one adaptation among many. It is the reason vertebrates can be as large and as active as they are. Oxygen dissolves poorly in water, and it dissolves poorly in plasma. If all you had was dissolved oxygen in a watery fluid, the amount your blood could carry would be pitiful, and the size and activity level of your body would be capped by that number. Hemoglobin breaks the cap. It is a protein that binds oxygen molecules and hands them off where they're needed, and it multiplies the carrying capacity of blood by a factor that transforms what a body can do. Every fish, every amphibian, every reptile, every bird, every mammal, from a hummingbird to a blue whale, runs on it, except 16 species of fish in the Southern Ocean. That number is worth holding on to. Not one weird species, an entire family, every member of it, all sharing the same absence, which tells you immediately that this did not happen 16 times. It happened once, in a shared ancestor, and every descendant inherited the whole. The genome shows the wreckage. Vertebrate hemoglobin is built from two different globin proteins that fit together, alpha and beta, coded by neighboring genes. In these fishes, the beta-globin gene is gone, not switched off, physically deleted from the chromosome. And what remains of the adjacent alpha-globin gene is a fragment, a partial sequence broken at the front end, the genetic equivalent of a page, torn out of a book with a few lines still clinging to the spine. It can't be read into a working protein, and it hasn't been for a very long time. One species in the family is a little different. It still carries a complete-looking pair of globin genes sitting next to each other, which for a while raised the possibility that this one animal had held on to the machinery. It hasn't. The sequence is riddled with the kind of damage that stops a gene from being expressed. It's a fossil in the genome, preserved well enough to show the shape of what was lost, dead enough that it produces nothing. More recent work points at how the deletion happened in the first place, and the answer is not romantic. These genomes are loaded with transposable elements, stretches of DNA that copy themselves and jump around, and they sit precisely at the boundaries of the missing region. When two copies of the same mobile sequence end up flanking a gene cluster, the cell's own repair and recombination machinery can misalign them and snip out everything in between. That is a well-understood accident. It is not a targeted improvement. It's the molecular equivalent of a filing system deleting a folder because two labels looked identical. So the most extraordinary feature of this animal, the thing that makes it unique among vertebrates, may have started as a copy-paste error. Now here's where the easy story wants to take over, and it needs to be stopped early. The easy story is that this doesn't matter much because the fish is a cold water bottom dweller that barely moves, so it hardly needs oxygen anyway. Slow metabolism, cold body, low demand, problem solved. It's a comfortable explanation. It's also demonstrably too simple. Start with the size of the deficit. Without hemoglobin, the only oxygen in the bloodstream is what will physically dissolve into the plasma, and plasma is not good at holding gas. The carrying capacity of icefish blood works out to a small fraction of what a red-blooded relative from the same waters can carry, roughly a tenth, depending on the species and the conditions you measure under. That is not a modest handicap. It is the removal of about 90% of the oxygen delivery system, while the tissues on the other end continue to demand oxygen at the same rate as before. And the tissues do demand it. These are not sponges. Icefish are predators. They eat krill and small fish, and taking either one means catching something that is actively trying not to be caught. Some of them are large, over half a meter long, with a substantial mass of muscle to keep supplied. They are not immobile, they are not miniature, and they do not have the option of anaerobic living. Whatever their metabolic rate is, it is not zero, and the difference between what they need and what dissolved plasma oxygen can deliver has to be made up somewhere. The second problem with the easy story is that the fish's own body contradicts it. If demand were genuinely trivial, you would expect a stripped-down, minimal cardiovascular system, small heart, modest vessels, a quiet, low-output plumbing loop suited to an animal that doesn't ask much. Open one up, and you find the opposite. The heart is huge. Relative to body mass, an icefish ventricle can be several times the size of the equivalent organ in a closely related fish, from the same water that still has hemoglobin. It sits in the body cavity looking like it belongs to a much larger, much faster animal. The blood volume is inflated too, in some species up to around 4 times that of a typical bony fish. The vessels are wide. The capillaries are unusually large in diameter. The whole circulatory system has been enlarged, and enlargement in biology is never free. That is not the anatomy of an animal that stopped needing oxygen. That is the anatomy of an animal that has been forced into an expensive workaround. There's a third piece, and it makes the situation stranger rather than simpler. Hemoglobin isn't the only oxygen-binding protein vertebrates use. Muscle tissue, especially heart muscle, muscle there, typically carries myoglobin, a related protein that holds oxygen inside the cells and buffers the supply so the muscle can keep contracting when delivery dips. It's the reason heart muscle and hard-working swimming muscle are dark. Several icefish species have lost that too. Their heart muscle contains no myoglobin at all. And this loss is not a single shared event like the hemoglobin deletion. Across the family, myoglobin expression appears to have been knocked out independently more than once, in different lineages, by different genetic mechanisms. Which is the detail that should make you uneasy. Losing hemoglobin once could be dismissed as a freak accident that the animals happened to survive. Losing a second oxygen-handling protein, repeatedly, in separate branches of the same family, in the same ocean, is a pattern. Something about this environment makes it possible to throw away equipment that is mandatory everywhere else on Earth. And whatever that something is, It has done it more than once. Species that kept myoglobin in the heart show measurably better cardiac performance than the ones that lost it, which tells you the loss carries a real cost and is not being compensated to indifference. The animals that lack it are running with a thinner margin. They manage. They are not thriving because of the absence. That distinction is going to matter enormously later, so it's worth putting plainly now. Nothing about this is an upgrade. This family did not evolve clear blood as a clever solution to living in the cold. There is no advantage to carrying 10% of the oxygen you used to carry. It is a wound inflicted by a genetic accident, and everything else in the body of these fishes is best understood as the long, expensive process of covering for it. Which raises the immediate question: how do you cover a 90% shortfall in oxygen delivery and stay alive, let alone hunt? There are only a handful of levers available, and the icefish pulls almost all of them at once. The first is volume. If each unit of blood carries a tenth as much oxygen, you can partly compensate by moving far more blood past the tissues in the same amount of time. That's what the oversized heart is for. Weight for weight, these fish push out roughly 4 to 5 times the cardiac output of a red-blooded fish of similar size in the same water. The pump is not just large for show. It is running an enormous circulation because it has to cycle the entire oxygen budget of the body through the tissues over and over to extract enough. The second lever is resistance. Pushing that much fluid through a normal fish's vascular network would require dangerous pressures, and a heart can only generate so much. So the pipes changed. The vessels are wider, the capillary beds are larger and more extensive, and the resistance the heart works against is low. The system operates the way a flood channel does, rather than the way a fire hose does. Enormous flow. Gentle pressure. Fish hearts in general run at low pressure compared to mammals, and this one has been pushed further in that direction, trading force for throughput. The third lever is the fluid itself, and this is where the cold quietly does the animal a favor. Cold makes fluids thicker. That's true of oil, it's true of syrup, and it's true of blood. Every degree you drop, the molecules move more sluggishly past one another and the fluid resists flow more strongly. For a fish living at -1.9, that is a serious mechanical problem. Ordinary vertebrate blood is not simply a liquid, it's a suspension, and the thing suspended in it is a dense population of red cells. Pack a fluid with cells, and you make it noticeably thicker. Chill that fluid to the freezing point of seawater, and you get something the heart has to fight. Icefish blood has almost nothing suspended in it. The proportion of the blood volume occupied by cells is effectively at rock bottom, close to nothing compared with any other vertebrate, which means that at the temperature where a red-blooded fish is pushing cold, cell-heavy sludge through its capillaries, the icefish is pushing something much closer to cold water. That is the one genuine consolation prize in this entire arrangement. The loss that destroyed the animal's oxygen capacity also stripped out the main source of blood viscosity, and it did so in the one environment on Earth where viscosity is at its most punishing. It doesn't come close to paying for what was lost. It does make the workaround physically possible, because driving that much thick, cold blood through an enlarged vascular network would be beyond what any fish heart could sustain. The blood is not frictionless, though, and there's a small irony buried in what makes it thick. With the cells gone, a significant share of what resistance remains comes from the dissolved proteins, and the antifreeze glycoproteins are a substantial part of that load. The molecules keeping ice from growing inside the animal are also, in a modest way, part of what its heart has to push against. The two survival systems are not independent. They're negotiating with each other in every capillary. The fourth lever is surface. If you cannot carry oxygen efficiently, you can at least be greedy about collecting it, and generous about handing it over. The gills are large, with a big total surface area of thin filament exposed to water that is close to saturated with dissolved oxygen. And then there's the skin. Icefish have no scales at all. And their skin is thin and richly supplied with blood vessels, which means it functions as a supplementary gas exchange organ. A meaningful fractal fraction of the animal's oxygen uptake appears to come straight through the body surface, an option available to it precisely because it lives in cold, oxygen-rich water, and doesn't have to worry about the water being warm and gas-poor the way a tropical fish does. Where a scaled fish has armor, this one has an extra lung. Put it all together and you get a specific, coherent machine. Take the oxygen carrier out of the blood. Compensate by inflating the heart, expanding the blood volume, widening the plumbing, dropping the resistance, exploiting the low viscosity that the missing cells left behind, and absorbing gas through every surface available. It works. The animals are alive. They hunt, they grow to substantial sizes, and they have held their place in this ecosystem for millions of years. And it is not elegant. It's brute force. The bill for it shows up in the energy budget. Circulating that much blood costs power. And estimates of the fraction of an ice fish's total metabolic output that goes into simply running the heart come out unusually high compared to other fishes. A larger slice of everything the animal eats is spent moving fluid around. That's energy not going into growth, not going into reproduction, not going into escaping a predator. It also caps what the animal can do in an emergency. A red-blooded fish under sudden demand has a reserve to call on. It can raise its heart rate, increase the oxygen extracted from each unit of blood, and lean on the pigment to buffer the surge. The icefish is already running its circulation near the top of its range just to sustain ordinary life. There is not much headroom above ordinary. It can accelerate, it can strike, it can push hard for a few seconds, and then it has to stop and recover because the system that supplies it has very little left in reserve. So this is an animal living permanently close to its own ceiling, in the coldest ocean on the planet, carrying a genetic injury it cannot repair, holding ice inside its body that it cannot dissolve. Which brings up the question that this whole picture has been building toward. Why is it still here? Natural selection is not sentimental. An individual born without hemoglobin should die, not eventually, immediately in the first generation, outcompeted by every sibling that still has it. There should be no lineage. There should be no family of 16 species. The mutation should have been erased the moment it appeared, the way thousands of catastrophic mutations are erased every generation without leaving a trace. The tempting answer is that it must have been good for something, that there was a hidden advantage, that clear blood somehow suits polar life, that this was selection producing a specialist. That framing feels right because it matches how we usually tell evolutionary stories, and researchers working on these fish have pushed back on it hard for decades. The phrase used in the literature is blunt. This looks like a loss that was tolerated, not a feature that was selected, a disadaptation, not an adaptation. The animals are not better off. They are getting away with it. The distinction is not academic. It changes what you're looking at. If clear blood were an adaptation, the story would be about a clever fish. If it's a tolerated loss, the story is about an ocean that stopped enforcing the rules. And that is the direction the evidence points. Consider what has to be true for a hemoglobin deletion to be survivable at all. First, the water has to be extraordinarily rich in oxygen. Gas solubility in water goes up as temperature goes down, and it goes up substantially. Cold water simply holds more dissolved oxygen than warm water does, which is why a mountain stream can support fish that would suffocate in a warm pond. The Southern Ocean is not only cold, it is also violently mixed by wind and storms, and continually ventilated by dense water sinking off the shelf so its oxygen content sits near saturation from top to bottom. An animal there is swimming through the most oxygen-loaded seawater on the planet. The plasma-only system is pathetic at carrying oxygen, but it is at least being asked to load from the richest possible source. Second, the animal's demand has to be low, and cold takes care of that as well. A fish is ectothermic. Its body temperature is the water's temperature, and biochemical reaction rates fall steeply as things get colder. At minus 1.9, every enzyme in the body is running slowly. The whole animal ticks over at a metabolic rate far below what an equivalent fish in temperate water would require. That does not mean demand is trivial, and the oversized heart is proof that it isn't. It means the gap between supply and demand, while still severe, is narrower here than it could be anywhere else. Third, the temperature has to be stable, not just cold, constant. A fish that can barely keep up with its oxygen demand cannot afford a heat wave, because warmth raises metabolic demand and lowers the oxygen content of the water at the same time, squeezing the animal from both ends simultaneously. The southern ocean shelf has been thermally boring for millions of years, varying by perhaps a degree or two through the annual cycle in most places. Nothing about that ocean asks a fish to cope with a surprise. Fourth, the competition has to be weak or absent, and that last condition is the one that requires explaining how this ocean got made. Antarctica was not always a freezer. For most of its history, it was attached to the other southern land masses, part of a single enormous continent, and the fossil record from that period describes forests, rivers, and a climate that supported large land animals. The sea around it was connected to the rest of the world's oceans and behaved like a normal ocean, with warm water arriving from the north and heat spreading southward. Then, the continent finished separating. South America pulled away, opening the gap now called the Drake Passage. Australia pulled away on the other side. For the first time, there was an uninterrupted ring of open water at high southern latitude, with no landmass anywhere on the circle to block a current. Water in that situation does something specific. Driven by the strongest sustained westerly winds on the planet, it starts moving eastward, and it never has to stop because there's nothing in the way. What formed was the Antarctic Circumpolar Current, a band of ocean circling the entire continent, moving more water than any other current on Earth. The precise timing is a live argument. The gateways opened over a long window. The geological evidence gives different answers, depending on which proxy you trust, and whether the current came together tens of millions of years ago in something like its modern strength, or built up more gradually over a longer period, is genuinely unsettled. What is not in dispute is the effect. The current works as a wall. Warm water from the north can no longer flow directly to the Antarctic coast because it runs into a fast-moving belt of cold water going the wrong way and gets deflected. Heat that used to reach the continent stops arriving. The water inside the ring loses heat and has no way to replace it, so temperatures fall and keep falling. Ice builds on the land, then extends onto the sea, then becomes permanent. The shelf water settles into the condition it's in today, near its own freezing point, saturated with oxygen and locked in. The wall is not only thermal, it is biological. A current like that, combined with a steep temperature gradient across it, is extremely difficult for marine animals to cross. Larvae drifting south hit water they cannot develop in. Adults ranging south hit conditions their enzymes cannot handle. The Southern Ocean became a room with the door shut, and for the animals already inside, the cooling was an extinction. The fish fauna that lived on the Antarctic shelf before this happened was a normal, diverse assortment of marine fishes, the kind you would find on any temperate continental shelf, with different families filling different roles. That fauna is largely gone. As the water went below the freezing point of ordinary fish body fluids and ice became a permanent presence, animals without a way to stop crystal growth were removed from the shelf. Not outcompeted. physically eliminated by a phase change. What survived that filter was the group that had, by whatever route and whatever timing, the anti-freeze solution. And what they inherited was a continental shelf the size of a continent with almost nothing living on it. That is the setting in which a hemoglobin deletion becomes survivable. Picture the same mutation appearing in a fish on a temperate reef. Warm water, holding less dissolved oxygen, a metabolic rate several times higher. Predators that are fast and abundant. Competitors occupying every niche, all of them carrying a full oxygen delivery system. That animal is dead within a day, and its lineage ends with it. Now put the identical mutation into a fish in an oxygen-saturated ocean at minus 1.9 degrees, with a metabolic rate held down by the cold, in a habitat where most of the previous occupants have already been killed off by ice, and the survivors are its own relatives. The mutation is still bad. It still costs the animal enormously. It just isn't fatal anymore, because nothing in the environment is fast enough, or hungry enough, or efficient enough to exploit the weakness before the animal reproduces. The environment did not favor the loss, it stopped punishing it. There is a real debate layered on top of this, and it deserves to be kept open rather than flattened. One line of thinking holds that the loss was close to neutral in those conditions, a genetic accident that drifted into fixation in a small population simply because it wasn't being selected against strongly enough to be removed. Another argues there may have been some marginal benefit in the specific context of near-freezing water, most obviously in reduced viscosity, that helped it along. And a third strand looks at the downstream consequences and asks whether the compensations themselves were driven by something more specific than general need. That third idea is worth a moment because it's genuinely clever. Hemoglobin and myoglobin do not only bind oxygen, they also mop up nitric oxide, a small signaling molecule that, among many other jobs, tells blood vessels to relax and stimulates the growth of new ones. Remove both proteins from an animal, and you have removed a major sink for nitric oxide, so its concentration in the tissues should rise. Elevated nitric oxide would push exactly the changes these fish display: wider vessels, denser capillary networks, an enlarged heart. Under that hypothesis, the icefish's dramatic cardiovascular remodeling isn't a series of separate adaptive steps at all. It's a cascade, triggered automatically by the same loss that caused the problem. With the body's own signaling chemistry building the workaround, it's a hypothesis, and it's still being tested, and it may turn out to explain part of the picture rather than all of it. What makes it worth telling is what it implies about how this animal came to be. Not a designer, not a clever solution. A broken system whose breakage happened to trip a signal that partly rebuilt around the break, in the one ocean on Earth where the animal could survive long enough for that to matter. So the reframing here is total, and it's the hinge of everything that follows. Stop thinking of the icefish as an animal that evolved something remarkable. Start thinking of it as an animal that lost something essential in a place that let it get away with the loss, and then spent millions of years building scaffolding around the whole, which raises an obvious follow-up. If this ocean was permissive enough to allow the deletion of hemoglobin, what else did it allow these fish to lose? The answer is sitting in their skeletons, and it's older than the hemoglobin problem. Every fish faces a basic mechanical difficulty. Flesh and bone are denser than seawater, so an unassisted fish sinks. Most bony fishes solved this a very long time ago with a swim bladder, a gas-filled sac in the body cavity whose volume can be adjusted to make the animal's average density match the water around it. Get it right, and the fish hangs motionless in mid-water at zero energy cost. It's such an effective piece of equipment that it is close to universal in the group, and it is the main reason the open ocean is full of fish that appear to be doing nothing at all while remaining exactly where they are. The notothenioids don't have one, not a reduced one, not a modified one. Their ancestors lost it before this story starts, and no member of the group has re-evolved it. for a bottom-living fish, that is not a problem at all. If your job is to lie on the seabed and ambush whatever wanders past, being heavier than water is convenient. It keeps you planted. It stops the current from rolling you around. The ancestral nototheniod was, as far as the anatomy suggests, exactly that kind of animal. A heavy-bodied bottom-sitter with big pectoral fins for propping itself up and a mouth built for grabbing. The trouble starts the moment there's an opportunity above the seafloor. And in the newly emptied Southern Ocean, there was an enormous one. The water column over the Antarctic shelf is deep, cold, oxygen-rich, and, after the extinction of the previous fauna, close to unoccupied by fish. There were swarms of krill in it. There were copepods. There was a whole vertical dimension of food that nothing was eating, because the animals that would normally have exploited it were gone, and the survivors were stuck on the bottom by their own density. To get up there and stay there without a swim bladder, a fish has only two options: swim constantly, which costs energy it can't spare, or change what it's made of. They changed what they're made of, from two directions at once. The first is subtraction. Bone is dense, considerably denser than seawater, and a normal fish skeleton is a significant part of why the animal sinks. So the skeleton was allowed to stay soft. Across the group, the more buoyant species show reduced mineralization, bones that never fully harden, larger proportions of cartilage retained into adulthood, and in some cases a persistent notochord, the flexible rod that most vertebrates replace with a proper backbone during development. In effect, these fishes never finish building their skeletons. They stop partway and keep the lightweight draft version for life. The second is addition. Lipid is less dense than seawater, so storing fat provides lift. The most buoyant notothenioids carry substantial deposits of it in the tissues, along the body, and in some species in dedicated sacs, and it works well enough that a few of them come remarkably close to neutral buoyancy, within a fraction of a percent of the density of the water they live in. That's not a bottom fish that can occasionally rise. That's a fish that hangs in open water indefinitely without a gas bladder, doing it with soft bones and stored oil. And the group did not do this once. Different lineages arrived at intermediate degrees of buoyancy independently, producing a spread from heavy bottom dwellers through species that hover just above the seabed to fully pelagic midwater animals. It's the same solution applied at different intensities, in parallel, by relatives working the same problem from different starting depths. Now look at what it cost them, because this is the same trade appearing again in a different currency. A skeleton with low mineral content is a weak skeleton. It provides less protection against impact and predators, and it gives muscles a less rigid frame to pull against, which limits the force that can be delivered in a burst of acceleration. Cartilage is springy, where bone is stiff, and springiness wastes energy that should be going into thrust. The lipid solution has its own bill. Fat is buoyancy, but fat is also stored food. And a fish that must keep a large lipid reserve just to avoid sinking cannot metabolize it in a lean season without slowly becoming heavier. Its emergency fuel and its flotation are the same substance. Burn one and you lose the other. In a productive season, that's fine. In a bad one, an animal that draws down its reserves starts sinking away from the water where it was feeding, which makes feeding harder, which draws down the reserves further. There's also a depth constraint hiding in it. A gas bladder is adjustable, which is what makes it powerful and also what makes it dangerous, since gas volume changes with pressure, and a fish that ascends too fast can find itself in serious trouble. Lipid and light bone are not adjustable at all. The buoyancy is baked into the body's composition and can only be changed slowly over weeks by growing or consuming tissue. What these fish get in exchange is indifference to pressure. They can move up and down the water column freely without any gas volume expanding or compressing inside them, because there is no gas anywhere in the body. In an ocean where the shelf is deep and animals routinely traverse hundreds of meters of water, that turns out to be a quietly enormous advantage that no swim-bladdered fish enjoys. So the pattern repeats. Every solution in this animal is a workaround built out of what was already available, and every workaround that leaves a permanent structural weakness that the environment is not currently exploiting. And this is the point where a single family stops being the story and the whole ocean becomes the story. Because the fish that hollowed out its skeleton to reach midwater was not doing it in a crowded sea. The standard picture of a polar ocean is emptiness. Ice, wind, gray water, nothing living in it. That picture is wrong in a way that matters. The Southern Ocean is one of the most productive marine systems on the planet during its short, ferocious summer, when continuous daylight and nutrient-rich, upwelled water trigger phytoplankton blooms that feed swarms of krill dense enough to change the color of the sea from the air. Whales come from the other side of the world for it. The system is not poor. It's seasonal, extreme, and enormously concentrated. What it lacks is diversity of fish. On a temperate or tropical shelf, the fish fauna is a patchwork of unrelated families, each with its own evolutionary history, sharing out the available roles. Antarctica does not look like that. On the high-latitude shelf, the notothenioids are not one group among many. They are effectively the whole fauna. In the coldest regions, they account for something close to 3/4 of the fish species present and well over 90% of the total fish biomass. In terms of sheer weight of fish flesh in the water, this is a single lineage ocean. Take a moment on how unusual that is. It is not normal for one related group to hold that share of an entire continental shelf ecosystem. It happens on isolated islands, in lakes cut off from rivers, in cave systems, in places where a founder arrives, the door closes behind it, and everything that follows descends from that one arrival. It is the signature of isolation. And in this case, the isolation was created by the same current that killed the previous fauna. So from a handful of surviving bottom dwellers, one lineage radiated outward to fill every job that was going. Some stayed heavy and benthic, sitting on the sediment with their pectoral fins spread, taking amphipods and worms and whatever passed within reach. Some became the mid-water hunters that the lightened skeletons made possible, feeding on krill in open water. One became a small, silvery, permanently pelagic species that swarms in enormous numbers and functions as the ocean's main forage fish across huge areas of the shelf, the link that moves energy from krill up to everything larger. And some became large predators, including the toothfish that grows to well over a meter and hunts in deep water. The crocodile ice fishes fit into that picture as active predators, mostly taking krill and smaller fishes, spread across depths from the shallow shelf to several hundred meters down, which means the family with the missing hemoglobin is not a marginal curiosity clinging to the edge of the system. It is embedded in the middle of the food web, doing a job that requires it to catch moving prey. And that matters far beyond the fish themselves because of what eats them. Follow the energy upward, and the shape of this ecosystem becomes uncomfortably narrow. Sunlight feeds algae, including the algae that grow on the underside of the sea ice itself, in a thin brown layer that gets released into the water when the ice melts. Algae feed krill and copepods. Krill feed everything. And in the band between krill and the large air-breathing predators, the animals doing the transfer are overwhelmingly notothenioids. Weddell seals hunt them under the fast ice, diving into water at the freezing point and coming back up to breathing holes they maintain by grinding at the edges with their teeth. Emperor penguins take them, along with krill and squid, on dives that go far deeper and last far longer than a bird has any business managing. Adelie penguins take the smaller ones. Antarctic toothfish, themselves notothenioids, hunt the others. Seabirds strip them from the surface layers. Even the ice itself participates, because the pelagic species that concentrate under it use the platelet layer as cover and a nursery. So the survival of a family of fishes with damaged blood is not a footnote about strange biology. It is load-bearing. A very large fraction of the vertebrate life people associate with Antarctica is standing on a lineage that has no hemoglobin in one branch of it, no swim bladder in any branch of it, ice held permanently in its tissues, and a genome full of things that were lost rather than gained. That is the position this group occupies. Now look at what it has to do to stay there. Reproduction sounds like the boring part of an animal's life. In this ocean, it is the part where all of the accumulated compromises come due at once. Cold does not just slow down an adult fish, it slows down development, and it does so brutally. Every process inside a fertilized egg, cell division, the folding of tissues, the growth of organs, the burning of yolk, runs at a rate governed by temperature. Drop the temperature towards zero, and the whole schedule stretches. An egg that would hatch in days in warm water takes weeks in cool water and months down here. That single physical fact reorganizes the entire reproductive strategy of these fishes. Start with the eggs themselves. If an embryo has to survive for months before it can feed itself, it needs a fuel supply that will last for months. So the eggs are large and heavily loaded with yolk. Nototheniod eggs are among the biggest produced by any bony fishes, several millimeters across, dense pale spheres that look less like fish eggs and more like small pearls. That size is not decorative. It is a battery, sized for a very long charge. Large eggs mean fewer eggs. A female fish has a finite body cavity and a finite energy budget, and every egg she makes larger is an egg she cannot also make more of. Where a temperate fish might broadcast hundreds of thousands or millions of tiny eggs into the water and rely on sheer numbers, an Antarctic nototheniod produces a fraction of that. The strategy has shifted from mass production to individual investment. And once you have invested that heavily in each egg, abandoning them stops making sense. A fish spawning millions of eggs can afford to lose almost all of them. A fish spawning a few thousand cannot. So this group does something relatively unusual among fishes. Many of them guard. Remotely operated vehicle surveys have found ice fish sitting on nests on the seafloor, in shallow excavated depressions roughly a meter across, with a compact mass of eggs in the middle. The guarding fish, apparently males in the cases observed, do not leave when a large machine with lights approaches. They stay in contact with the eggs and switch into defensive behavior instead, which is not the response of an animal casually resting near a clutch. Larval size evidence suggests these eggs may sit there developing for 6 months or more. Think about what that commits the parent to. Half a year, or something in that range, spent stationed on a patch of mud in freezing water, defending a fixed point, unable to follow food, exposed to anything that comes past. In an animal already running its cardiovascular system near its limit just to stay alive, that is an extraordinary bill to accept. They accept it because the alternative is worse. In an ocean where an egg takes half a year to hatch, an unguarded clutch is simply a slow-release meal for everything that lives on the bottom, and there are plenty of those. Sea stars, ribbon worms, amphipods, other fish. Guarding is not devotion. It's arithmetic. Now scale it up. In the southern Weddell Sea, a towed camera system was dragged across the seafloor beneath the water near a great ice shelf, in a deep trough that funnels water in from the continental slope. The plan was straightforward seabed survey work. What the cameras recorded was a nest. Then another nest, then another, and the pattern did not stop. For hours of towing, across kilometers of seabed, the same image kept repeating. A shallow circular depression in the mud, a pale rim of small stones, a clutch of eggs, and a single ice fish sitting on it. Then another one, a couple of meters away. then another. The survey mapped nests at a density of roughly a quarter of a nest per square meter across an area estimated at around 240 square kilometers, and the extrapolation from that came out at something on the order of 60 million active nests, with the fish biomass associated with them running into tens of thousands of tons. It is the largest continuous fish breeding colony documented anywhere on Earth, at any depth, and it was sitting under Antarctic ice, unknown, until a camera happened to be towed across it. Take a second with the image itself, because it is one of the strangest things a camera has ever recorded on a seafloor. Not a shoal, not a swarm, a grid. Millions upon millions of individual excavated bowls, spaced out with rough regularity like plots on an allotment, each one occupied by a single pale fish lying over its eggs in permanent darkness, with nothing above it but black water, and far beyond that, ice. And every one of those animals is a body with no hemoglobin, holding a defensive station for months in water at its own freezing point. There are questions attached to it that have not been answered. Why there? The best available explanation points at water properties. The site sits where a slightly warmer, deeper water mass rises up onto the shelf, and that particular water may create conditions that suit the eggs, or that concentrate the food supply, or both. Whether the colony is permanent, whether it reforms in the same place every year, how long it has been there, and how many similar aggregations exist elsewhere under ice, where no camera has ever been towed, are all open. One survey line across a continental shelf is a very thin sample of a very large area. What the colony does establish is a link that the rest of the ecosystem was already exploiting. Tracking data from Weddell seals in that region showed the animals spending significant time in the water above the colony, which is not a coincidence anyone needs to strain to explain. 60 million fish sitting still in one place, unable to flee without abandoning their entire reproductive investment, is not a hiding place. It's a buffet with a fixed address. And that is the vulnerability this whole strategy carries. Slow development does not stop at hatching. It runs through the entire life of the animal. A larva that emerges from one of those eggs is comparatively large and well developed, which is the payoff for all that yolk, and it is still facing years of growth at temperatures that make growth expensive and slow. Antarctic notothenioids are late maturers. Depending on the species, a fish may need several years, in the larger predators a decade or more, before it reproduces at all. They grow slowly, they mature late, and they can live a long time, which is a coherent strategy in a stable environment and a dangerous one in an unstable one. Here's why that combination is fragile. A fish that matures in 1 year and spawns 1 million eggs can rebuild a devastated population within a few seasons. Knock the numbers down and the survivors flood the system with offspring, and the population springs back. A fish that takes 8 years to mature and produces a few thousand large eggs, most of which are guarded by a parent that cannot guard them and feed at the same time, has no such capacity. Remove a large fraction of the adults, and the recovery timescale is measured in decades, assuming conditions stay favorable for that entire period. There's an additional weak point at the very start of life that is not fully understood. Adult notothenioids are loaded with antifreeze. Larvae are not. Measurements on early-stage fish show antifreeze levels far below adult concentrations, which raises an obvious problem, because a larva in shallow Antarctic water is swimming in the same ice-laden environment its parents are. How they manage that gap is not settled. Part of the explanation may lie in where the larvae spend their first months and how much contact they actually have with ice, and part of it may be that a small, smooth, not yet feeding animal simply offers ice fewer entry points than a large one that drinks seawater constantly. What is clear is that the freezing defense that defines this group is not fully in place at the stage of life when the animals are most numerous and most vulnerable. And then there's the detail that turns the whole freezing problem inside out. The platelet ice layer, the mesh of loose crystals hanging in sheets beneath the fast ice. is the single most dangerous place in this ocean for an animal that can be nucleated. It is a three-dimensional tangle of ice, meters thick in places, in direct contact with the water that fish are swimming in. By every argument made so far, it should be the one part of the system that vertebrates avoid completely. It is a nursery. Juveniles of several nototheniod species concentrate in and around that sub-ice layer, and at least one small member of the group has gone further and made it a permanent address. It lives up against the ceiling rather than on the floor, moving through the gaps in the platelet mesh, often oriented with its belly toward the ice above it, feeding on the copepods and small crustaceans that gather in the same layer. Think about what that animal is doing. It is spending its entire life in physical contact with ice crystals, in water at the freezing point, relying on nothing but adsorbed protein to keep those crystals from seeding it. There is no distance between it and the thing that would kill it. The margin is a molecular film. Why do it? For the same reason anything accepts a hostile address, the alternative is worse. The platelet layer is structurally complex, and complexity is cover. A small fish inside that mesh is difficult for a Weddell seal to chase down, and the seals hunting beneath the fast ice are fast, large, and extremely capable in open water. It is also close to the food that accumulates at the ice-water boundary, where the algae are, so the ice is simultaneously the lethal hazard and the safest room in the house. And the antifreeze is what makes it possible to choose the second reading over the first. That inversion is worth holding on to because it captures the whole logic of this ocean. Every advantage here is an accepted hazard. The cold that would kill an ordinary fish is what keeps the water oxygenated. The ice that could nucleate a body is what shelters the young. The isolation that removed all competition is the same isolation that removes any escape route. So the picture of this lineage is now complete enough to state plainly, and it should be uncomfortable. These are animals with almost no oxygen-carrying capacity in one branch of the family, running an oversized heart at close to full output just to sustain ordinary life, holding ice in their tissues that they cannot melt, floating on soft bones and stored fat because they threw away the swim bladder, breeding on the seafloor in colonies that cannot flee, guarding eggs for half a year, taking years to mature, and depending on antifreeze proteins that are precisely tuned to a temperature range they will never leave. Every one of those traits works. Every one of them is also a bet on the water staying exactly as it is. Which brings this to the part that actually matters. If you had to guess which vertebrates on Earth are the toughest, you would probably reach for the extremophiles, the animals that survive conditions that would kill anything else. Something that lives at the freezing point of seawater, under permanent ice, in the dark for decades, sounds indestructible by definition. Surviving Antarctica must require the biological equivalent of armor plating. That instinct is exactly backwards, and the data on it is not ambiguous. The standard way to measure a fish's heat tolerance is to warm the water at a steady rate and record the temperature at which the animal loses the ability to keep itself upright and coordinated, the point where its nervous system and muscles stop working together well enough to escape. That value gets called the critical thermal maximum. It's a crude measurement, and everybody who uses it knows it, because warming an animal quickly in a tank is not the same as warming an ocean over decades. As a comparison between species, though, it is useful. Run that test on fishes from temperate coasts, and you get values well up in the 30s. Run it on tropical reef fishes, and you get numbers in the same broad range. Run it on a range of Antarctic nototheniod, acclimatized to their own natural freezing point water, and the values come out roughly between 12 and 16 degrees Celsius. Now, 12 to 16 degrees does not sound like a fragile animal. That's a comfortable swimming pool temperature. It sounds like these fish have a huge amount of headroom over the minus 1.9 they actually live at. And if that were the real limit, warming would be a distant problem. It is not the real limit, and the gap between that number And the real limit is the whole story. A critical thermal maximum is a measure of acute collapse. It's the temperature at which the animal falls over during a fast ramp, not the temperature at which it can live. The temperature at which an animal can actually survive long-term, growing, digesting, maintaining tissue, producing eggs, and outrunning predators is far lower, because all of those processes fail long before the nervous system does. When Antarctic notothenioids are held at elevated temperatures for extended periods rather than ramped quickly, the picture changes dramatically. Chronic limits sit far below the acute ones, and the range of temperatures in which these fish can sustain normal function is startlingly narrow, in some species only a few degrees above the water they currently occupy. Push a notothenioid a handful of degrees above its natural range and hold it there, and it does not die immediately. It stops eating properly. It stops growing. Its metabolic costs rise faster than its ability to pay them. The animal is not killed, it is drained. The mechanism behind that squeeze is the same one that made this ocean permissive in the first place, running backwards. Warm water holds less dissolved oxygen. That's the first jaw of the trap. As temperature rises, the concentration of oxygen in the sea drops, so every liter of water passing over the gills carries less of what the animal needs. Warm tissue demands more oxygen. That's the second jaw. Metabolic rate in an ectotherm rises steeply with temperature, and the rise is not gentle. A few degrees can increase resting demand substantially before the animal has done anything at all with the extra energy. So warming reduces supply and increases demand simultaneously, and the animal is caught between them. Every fish on Earth faces some version of this. What makes the ice fish situation different is where it starts from. A red-blooded fish meeting that squeeze has a hemoglobin system with reserve in it. It can extract more oxygen per unit of blood. It can raise the proportion it unloads at the tissues. It can recruit capacity that was sitting idle. There is slack in the system, and slack is what gets an animal through a bad summer. An ice fish has no slack. It is already moving 4 to 5 times the normal volume of blood, already running a heart that is enlarged to the limits of what the body cavity and the energy budget will support, already relying on the water being nearly saturated with oxygen, already exploiting the low viscosity of cold blood to make the flow rates achievable. Every compensation has already been spent. There is nothing left to deploy, and when researchers have tested exactly that, warming these animals and measuring how hard the heart can work, the results line up with the prediction. Maximum cardiac performance in the hemoglobinless species falls off at lower temperatures than in their red-blooded relatives. The ones that also lack myoglobin in the heart muscle do worse still. The animals that gave up the most equipment are the first to fail when the water warms, which is precisely what you would expect from a system that was only ever viable because the environment was doing half the work. The failure does not stop at the heart. It reaches all the way down into the proteins. An enzyme is a machine made of a folded chain, and its job depends on that chain having exactly the right amount of flexibility. Too rigid, and it cannot change shape to grab its target. Too floppy, and it falls apart. Every organism tunes its proteins to the temperature it lives at, and the tuning is a compromise between those two failure modes. At minus 1.9 degrees, molecular motion is sluggish, and a normally structured protein becomes too stiff to do anything useful. So cold-adapted animals build looser proteins, with weaker internal bonds and more conformational give, so that at freezing temperatures they still have the mobility to function. That's a real and well-documented feature of polar biochemistry, and it's why these fish are not simply slow versions of temperate fish. Their machinery has been rebuilt to work in the cold. The cost is written into the same molecules. A protein that is loose enough to work near zero is a protein that comes apart easily when it warms. It has less internal bracing, so it unfolds at lower temperatures than the equivalent protein in a temperate animal. The exact adaptation that makes life possible at the freezing point is the adaptation that makes moderate warmth destructive, which is precisely where the next missing piece shows up. Every organism from bacteria upward has a defense for this. When cells get too hot and proteins start unfolding, they mount an emergency response, a rapid burst of production of a specific set of molecular chaperones, proteins whose job is to grab misfolding chains, hold them, and either refold them or escort them to be destroyed. It's called the heat shock response, and it is one of the oldest and most conserved cellular defenses known. Yeast has it, insects have it, you have it. It is not a specialist adaptation, it's basic vertebrate equipment. Antarctic notothenioids appear to have lost the inducible version of it. Heat a temperate fish, and the chaperone genes fire, transcript levels climb sharply, and the cell floods with protective protein. Heat these fish, and that surge does not come. The classic response, the sharp induction under thermal stress, is absent or drastically reduced in the species that have been examined. Under the kind of heat that should trigger a full emergency, the overall transcriptional pattern tends toward shutting down rather than mounting a defense. The genes are not gone. What appears to have changed is the regulation. The chaperone that is supposed to be the emergency reserve is instead produced continuously, at high baseline levels, all the time, which makes a certain grim sense because these animals live with a chronic protein problem. Cold-adapted proteins are unstable by design, and Antarctic fishes carry unusually high levels of damage-tagged protein waiting for disposal. Their cells are not idling. They are running permanent maintenance on machinery that is always slightly falling apart. The emergency system got promoted to full-time janitor, and in the process, the ability to escalate was lost. That is the same pattern as the hemoglobin, expressed in a different currency. In an environment with no thermal surprises, a system for surviving thermal surprises is dead weight. Selection stopped maintaining it. The regulation drifted. And an animal that hasn't needed an emergency response in millions of years is now an animal that does not have one. There is a small qualifier here that deserves to be kept, because the story is not quite as absolute as it first looked. When these fish are given a sublethal warm exposure and then tested, several species show a modest improvement in their tolerance afterward, on the order of a degree or so. Some capacity for adjustment survives. A handful of species can also raise their upper limits somewhat if they are held at warmer temperatures for an extended acclimation period, and the ability to do this varies significantly between species, with some responding far better than others. So the door is not entirely shut. It is open by roughly a degree unevenly, in some species and not others, and against a physical driver that does not care about degrees of politeness. Now stack all of it up and look at the animal as a whole, because the individual losses are less important than the pattern they form. No hemoglobin in one branch, no cardiac myoglobin in several lineages, no swim bladder in all of them, no fully mineralized skeleton in the buoyant ones, no inducible heat shock response, no thermal reserve, no physiological slack, an oxygen delivery system built entirely on the assumption that the water will always be cold and always be saturated, a freezing defense tuned to a range a couple of degrees wide, a reproductive cycle that commits an adult to a fixed patch of seafloor for half a year and takes years to complete a generation. This is not a robust animal. This is an animal with every redundancy stripped out, running at the edge of its own capacity, in a habitat that has been almost perfectly constant for longer than our species has existed. And that is only a viable way to exist while the constant holds. Here is the part that people usually miss when they hear that polar animals are threatened by warming. The instinct is to picture the water heating up and the fish being cooked, and to reassure yourself with the fact that Antarctic water is very cold, and warming is measured in fractions of a degree per decade, so surely there's time. The threat is not really the thermometer. The threat is what the temperature is holding in place. Go back to the wall. The Antarctic circumpolar current is not just a band of water. It is the boundary condition for this entire biological system. It keeps warm northern water away from the continent, which keeps the shelf near freezing, which keeps the notothenoid enzyme set functional, which keeps the antifreeze proteins matched to their working range, which keeps oxygen levels high enough for a hemoglobinless fish to breathe. Every layer of the animal's biology is stacked on that one oceanographic feature. It also keeps other animals out. This is the part that gets underestimated. For millions of years, the cold has functioned as a barrier not just to heat, but to entire categories of predator. Certain groups of marine animals cannot function in near freezing water for reasons that come down to basic physiology. Crabs and lobsters and their relatives have difficulty regulating magnesium in their body fluids at low temperatures, which leaves them progressively unable to coordinate muscle activity as the water cools. Many sharks and their relatives face their own constraints there. Fast-swimming, hard-shelled, bone-crushing predators, the kind that dominate shallow seas everywhere else on Earth, have been largely absent from the Antarctic shelf for a very long time. Think about what that means for the animals living there. The shelf fauna evolved in a world without shell-crushing predators. Antarctic invertebrates are famously soft, thin-shelled, slow, and poorly defended by the standards of anywhere else. Sea stars, brittle stars, sponges, sea spiders of remarkable size, mollusks with fragile shells, huge slow-growing communities sitting exposed on open sediment. That entire assemblage exists because nothing has been able to come and eat it in the way things do at lower latitudes. The ice fish sitting on its nest for 6 months, unable to flee without abandoning its investment, is a member of the same assemblage. Its strategy makes sense in an ocean without an efficient benthic predator capable of working through a colony of stationary adults. That is not a permanent feature of the universe. It's a feature of the temperature. And the temperature is what's changing. Not uniformly, and not everywhere at once. The Southern Ocean is enormous, and it does not warm like a pan on a stove. Some regions have shown little change. The West Antarctic Peninsula, the long arm reaching up towards South America, has been among the more rapidly changing marine regions on the planet, with sea ice declining and water temperatures shifting over recent decades. Elsewhere, the more consequential change is not surface warming at all, but something quieter. Relatively warm, salty water from deeper in the ocean pushing up onto the continental shelf through the troughs that cut across it, flowing in beneath the cold surface layer and reaching the undersides of ice shelves. That deep water is warm only by Antarctic standards, a degree or two above freezing rather than at it. Against an ice shelf, a degree or two is an industrial-scale melting tool applied continuously for decades. Against a fish tuned to a 2-degree envelope, it is not a trivial number either. And it is already the water that some of these fish are living next to. The great nesting colony in the Weddell Sea sits where exactly this kind of deeper, slightly warmer water rises onto the shelf, which is very likely part of why it is there in the first place. The animals are using the edge of a water mass, which means changes in how that water mass behaves are not an abstract future problem for them. They are the local weather. Now put the second consequence next to it. In the deep water off the Antarctic Peninsula, surveys have documented populations of king crabs, large lithidid crustaceans, living on the continental slope, including a substantial reproductive population found in one deep basin. These are exactly the kind of animal the cold has excluded from the shelf. Generalist, powerful, capable of crushing and tearing invertebrate prey, and known elsewhere in the world as ecosystem restructuring predators. Their presence on the slope, at depths where the water is slightly warmer than the shelf above, puts them at the edge of a habitat full of soft-bodied, thin-shelled animals that have not faced anything like them in millions of years. Whether this constitutes an invasion is genuinely contested, And the disagreement is worth understanding because it changes the interpretation completely. One reading is that these crabs are newcomers, pushed onto the Antarctic margins as the thermal barrier weakens and poised to move shallower as the shelf warms. Another reading, argued from the distribution and the genetics, is that lithodids have been living in Antarctic deep water for a long time, that their absence from the shallow shelf reflects a boundary they have always been sitting against rather than a frontier they are currently crossing, and that reports of an ongoing invasion have run ahead of the evidence. Both readings agree on the physical fact underneath the argument. There is a temperature line, the crabs are on one side of it, an entire defenseless fauna is on the other, and the line is set by the water rather than by anything the fauna can control. That is the vulnerability that the whole of this story has been circling. The icefish is not fragile because it is delicate. It is fragile because it is precise. It is a body machined to fit a very specific set of conditions, with almost no tolerance left over. And the conditions are maintained by an ocean current and a temperature gradient, rather than by anything the animal does. The fish does not have a fallback. Consider what its options would be. It cannot move to colder water because there is none. This is already the coldest seawater on Earth. There is no further south to go, no deeper refuge that is colder than the shelf, no seasonal migration that leads anywhere better. Most temperate and tropical species facing warming can shift their range poleward, and that is precisely what many of them are observed doing. A fish that already lives at the pole, at the freezing point, has run out of map. It cannot adapt quickly because its generation time is measured in years, and its capacity for physiological adjustment is limited to about a degree in the species that have been tested. Evolution is not fast enough when a population takes 8 years to turn over, and the environment shifts within decades. It cannot rebuild its equipment. The hemoglobin genes are not switched off, they are deleted. That is a one-way door. There is no plausible route back to a red-blooded icefish because the sequence required to make the protein no longer exists in the genome to be reactivated. Whatever the future holds for this family, it will be faced with clear blood. And it is being pressed from another direction at the same time, which has nothing to do with temperature. The Southern Ocean is fished. Krill are harvested in quantity, concentrated in some of the same regions where the ecosystem is under the most thermal pressure, and krill is the foundation this entire food web is standing on. Antarctic toothfish and their Patagonian relatives support a valuable deep-water fishery, and the icefish themselves have been targeted directly. One midwater species was trawled heavily around the subantarctic islands in the decades when the Southern Ocean was being opened up commercially, and the stocks were driven down hard enough that recovery has been slow and uneven under subsequent management. The toothfish is the case that shows how far the consequences run. This is the giant of the group, a notothenioid that reaches well over a meter and a substantial weight. Neutrally buoyant on the same combination of soft bone and stored lipid as its smaller relatives, carrying the same antifreeze, and sitting at the top of the fish food web in deep Antarctic water. It eats other notothenioids. It is eaten in turn by sperm whales and killer whales, and, when it comes shallow enough, by seals. It grows slowly, matures very late, and lives a long time, which are the standard properties of a large cold water predator, and also the standard warning signs for anything that gets fished. It is fished. Toothfish are among the most valuable wild-caught fish in the world, sold under a market name that most consumers would never connect to Antarctica, taken on long lines set in deep water around the continent and the sub-Antarctic islands. The legal fishery operates under a management framework built specifically for the Southern Ocean, with catch limits and observers, and it has spent decades contending with illegal fishing that operated outside all of it. Set that against the biology, and the tension is obvious. A fish that takes a decade or more to mature In an ocean where growth is slow because the water is cold, cannot absorb the same removal rate as a temperate species that spawns at 2 years old. And unlike a cod stock in the North Atlantic, there is no adjacent population from a warmer region that can drift in and repopulate a depleted area, because the current that isolates this ocean isolates the fish inside it too. That last detail is a useful test of everything said earlier about slow life histories. A fish that matures late produces relatively few large eggs, guards them for months, and grows slowly in cold water is precisely the kind of animal that fishing pressure removes faster than reproduction replaces. The reproductive strategy that works beautifully in a stable ecosystem with no industrial predator is close to worst case when one arrives. There is a temptation at this point to slide into forecasting, and it should be resisted, because the honest answer about what happens next is that nobody knows in detail. Predicting how a specific fish population will respond to a shifting ocean requires knowing how fast the water will change in that particular place, how the food supply will respond, whether spawning grounds remain suitable, whether new predators actually arrive, and how much physiological flexibility the animals retain, and every one of those has real uncertainty attached. Projections exist. They disagree with each other in the details. What they largely agree on is the direction and the reason. These are among the most thermally specialized vertebrates known, in a region where change is not hypothetical, with unusually little capacity to relocate or adjust. The uncertainty is not comfort. Uncertainty about how fast a very narrow window is closing is not the same as evidence that the window is wide. And that window is not only a temperature range. It's also a calendar, and the calendar is written by ice. Everything living on the Antarctic shelf runs on a light supply that behaves nothing like the one most of the planet uses. Near the continent, the sun does not set for months in summer and does not rise for months in winter. Photosynthesis, which is where every calorie in this system originates, therefore does not tick along at a moderate rate year-round. It detonates and then stops. In the light season, with continuous sun, nutrient-rich water welling up from below, and the retreat of the ice opening the surface to the sky, the phytoplankton bloom is immense. There's a second production system layered on top of it that most people never hear about. The sea ice itself is alive. Algae grow inside the brine channels in the ice and in a dense layer on its underside, so that the ceiling of this ocean turns brown and slick with living cells. When the ice melts back, that layer is dumped into the water column in a concentrated pulse. Krill are built around that pulse. Adult krill graze the open water bloom in summer, and larval krill, in their first winter, depend heavily on the algae living on the underside of the ice, scraping it from the ceiling in a season when there is nothing else to eat. Sea ice is not just a physical structure to them. It's a pantry and a nursery combined. Then the light goes, and the entire system shuts down. For months, production collapses toward nothing. The bloom is gone. The water clears. The only energy left in the system is whatever was stored during the summer, either as fat inside animals or as organic material that sank to the seafloor and is being slowly worked over by whatever lives down there. Now put a fish with no metabolic slack into that cycle. It has two ways through the dark half of the year, and it uses both. The first is the cold itself, which drops its energy demand to a level that a temperate fish could not achieve if it tried. Slow enzymes, slow digestion, slow growth, slow everything. A body that is expensive to run in warm water is astonishingly cheap to run at the freezing point, and that cheapness is exactly what allows an animal to sit through months of scarcity without starving. The second is storage, and here the buoyancy solution turns out to be doing double duty. The lipid these fishes carry to keep from sinking is also the reserve they burn when there is nothing to eat. It is a fuel tank that doubles as a flotation device, and that is efficient right up until the moment the season runs long, at which point the animal is spending its buoyancy to stay alive and getting heavier as it does. There is no version of this where a bad year is cheap. The consequence is written into how these fish grow. Growth in the Southern Ocean is slow to the point of looking pathological by temperate standards. Individuals put on very little in a year, which is why maturity takes so long, which is why the reproductive turnover of the population is so sluggish, which is why removing adults from the system is so much more damaging here than it would be in a warmer sea. All of it traces back to a light cycle and a temperature, and every element of that supply chain is tied to sea ice. How much forms, how long it stays, when it retreats, and how much algae it carries. Change the ice, and you have changed the calendar that this animal's entire energy budget is built around, regardless of what the thermometer says about the fish itself. So how does an animal with almost no oxygen-carrying capacity, running its heart near maximum just to idle, actually catch anything in a system like this? The answer is that it cheats, and the way it cheats explains the shape of its body. An ice fish is not built to chase. Look at the proportions again. The head is enormous relative to the body, broad and flattened, with a mouth that opens wide and a skull built to expand rapidly. The body tapers away behind it, and the tail, while functional, is not the deep muscular wedge of a fish that makes its living on sustained speed. The pectoral fins are large, splayed, and set low, ideal for hovering, bracing on the bottom, and making fine adjustments in position, and useless for outrunning anything. This is the anatomy of an ambush predator. An ambush is the only feeding strategy that makes sense for a body with this power supply. The economics are straightforward once you see them. A pursuit predator spends energy continuously, over long periods, and must sustain high oxygen delivery to muscle for the entire chase. That is exactly what an ice fish cannot do, because its circulation is already at capacity. An ambush predator spends almost nothing for hours, and then spends a lot for a fraction of a second. The strike is powered largely by stored elastic tension and a burst of anaerobic muscle activity, which does not require oxygen at the moment of use, only afterward during recovery. A body with no reserve can still afford one violent instant, provided it gets to lie still before and after. The strike itself is suction. Open a large mouth fast enough, and the sudden expansion of the buccal cavity drops the pressure inside it, and water rushes in, carrying whatever was in front of it. Krill do not get chased down. They get inhaled. The wide head is not for biting hard, it's a pump. And the small teeth that line the jaws are not for cutting. They're for making sure that whatever came in with the water does not swim back out. Now add the environment, because it is stacking the deck in the predator's favor in a way that is easy to miss. The prey is cold too. Muscle contraction is a chemical process, and it slows in the cold exactly like everything else. Krill in water at -1.9 are not darting the way they would in warmer water. Their escape response is slower, their swimming speed is lower, their reaction time is longer. Every prey animal in this ocean is operating with the same handicap as the predator, which means that a slow predator is not automatically a failed predator. The whole system has been dialed down together. That is one of the quiet reasons a hemoglobinless ambush hunter is viable here, and would be unthinkable on a tropical reef, where prey animals accelerate at rates that demand real power to intercept. Cold does not just reduce the fish's need for oxygen, it reduces the performance requirement of the job it is trying to do. Then there's the problem of finding anything at all in the first place. For half the year, this ocean has no sun at all, and even in summer, the light that reaches a fish sitting 200 meters down beneath a meter of ice, and a layer of snow is a fraction of what falls on the surface. Snow is the critical part. Bare sea ice transmits a reasonable amount of light. Put snow on top and it reflects most of it back to the sky, so the water underneath a snow-covered floe can be startlingly dark in the middle of the polar day. The fish are built for that. The eyes are large and dark and take up a substantial share of the head, which is the standard signature of an animal working in dim light. A big lens and a wide aperture gather more photons, and a large retina behind them gives more surface for those photons to land on. It's the same solution deep-sea fishes use, arrived at for the same reason. And it works because there is usually some light down there. Not much. Enough to make out a silhouette against the faint glow filtering down from above. That geometry matters for how a bottom-sitting predator hunts. Looking up, prey is backlit. Looking down, prey is invisible against black sediment. So the ambush hunter on the seafloor is not scanning the ground. It is watching the ceiling, waiting for something to cross between it and what little light is coming through the ice. Vision alone would not be enough in a system this dark, and it isn't the only sense in play. These fishes carry a well-developed lateral line, the row of pressure-sensitive organs running along the flank and branching across the head that lets a fish detect water movement around it. A krill beating its swimmerettes, a small fish turning nearby, a disturbance in the water column, all of that produces a pattern of pressure changes that a lateral line can pick up without any light at all. For an animal that spends long periods motionless on the bottom, that's not a backup system. It's a proximity alarm covering the space it cannot see. There's a third source of information in this ocean that has no equivalent on most continental shelves. Krill produce their own light. They carry photophores that emit a soft blue glow, and swarms of them can light up in the dark for reasons that are still argued over, from social signaling to counter-illumination camouflage. Whatever the function, a predator sitting in blackness with large light-gathering eyes is being handed a signal by its own prey. Whether icefish actively hunt on that cue has not been demonstrated, and it should be treated as an open possibility rather than a fact. But the ingredients are all present: a dark environment, an animal that glows, and a predator built to collect faint light. So assemble the working animal, a body that hangs or rests without effort because it has traded bone for cartilage and fat. A circulation running several times harder than it should have to, because there is no pigment in it to carry oxygen, made possible by blood that is thin because it is nearly empty, and by water that is nearly saturated because it is nearly frozen. Antifreeze proteins in the fluids, holding captured ice motionless in tissues that will carry it for years. Enzymes rebuilt to work at temperatures where ordinary enzymes cease. Large eyes turned upward toward a ceiling of ice. A lateral line reading the water. A mouth that opens fast enough to inhale prey that is too cold to escape quickly. And a metabolic rate low enough to sit through half a year of darkness on stored fat. That is not a monster. It is a tightly integrated survival system with no loose parts anywhere in it, and every component is conditional on the water not changing. There's one point in the year when that system has to abandon its own logic, and it's the one already described. An ambush predator's entire strategy is built on being able to choose where to be and to stay hidden while it waits. A nest-guarding adult gives that up completely. It sits on a fixed patch of open sediment over a bright cluster of eggs for months, defending rather than hiding, unable to follow prey, and unable to retreat. Everything about that behavior contradicts the animal's own design. They do it because nothing else produces surviving offspring when the water is this cold, and development runs this slowly. It's the single largest gamble in the life cycle, and it explains why the colony under the Weddell Sea ice looks the way it does. Not a shoal seeking safety in numbers. Millions of individual animals, each making the same forced bet, in the same place, at the same time, because the conditions that make the bet survivable exist there and not somewhere else. And now widen the frame one more time, because the consequences of that bet do not stop with the fish. Almost every vertebrate in the Southern Ocean sits above the same narrow band of the food web. Krill and small crustaceans convert plankton into flesh. Notothenioids convert krill into larger packages of flesh distributed across depths, including into the deep water and the seafloor, where krill themselves do not spend most of their time. And then seals, penguins, seabirds, and larger fish take those packages, remove or thin out that middle layer, and there is no other lineage waiting to take over the job, because this ocean does not have a diverse fish fauna in reserve. The isolation that let one group inherit the shelf also means there is no bench. In a temperate sea, if one family of forage fish collapses, others move into the gap, because a dozen unrelated lineages are already present and competing. Here, the notothenioids are effectively the entire team. That is what makes this more than a story about an unusual animal. The strange physiology and the ecosystem risk are the same fact seen from two distances. The lineage was able to take over the shelf because the conditions were extreme and stable enough to exclude everything else. Those same conditions allowed it to shed equipment, which locked it into the narrow envelope it now occupies. So the group holding up the food web is also the group with the least capacity to tolerate change. And there is no replacement standing by. There are already signals worth watching, and they need to be handled carefully, because attribution in a system this large and this poorly sampled is genuinely difficult. The small pelagic notothenioid that serves as the main forage fish across much of the shelf has become scarce in parts of the Antarctic Peninsula region, where it was formerly a dominant component of the fish community. Whether that reflects warming, changes in sea ice affecting where its eggs and larvae can develop, shifts in the krill supply, natural variability, or some combination, has not been resolved. Krill distribution in parts of the Southern Ocean has also shifted, and the relationship between krill recruitment and winter sea ice extent is a genuine research question rather than a settled equation. What is not in doubt is the structure. A short food chain with few redundancies, standing on a fish lineage tuned to an unusually narrow set of physical conditions. In the part of the world where those conditions are set by ice, which raises the question of how much of this we are actually in a position to see. Consider what it takes to learn anything at all about these animals. The Southern Ocean is the most hostile working environment on the planet for a research vessel. The weather is violent, the season is short, the ice moves, and the ships capable of operating there are few and expensive. Most of what is known about Antarctic fish physiology comes from a handful of coastal stations where researchers can reach the water, and from ship time measured in weeks, not years. Even catching the animals is a specialized problem. Fish taken from the shelf have to be handled in water kept at their own temperature, because a bucket of seawater sitting in a heated laboratory is, for a notothenioid, a slow execution. Aquarium systems for them run permanently at around the freezing point. Anything warmer than a couple of degrees is stress, and anything much warmer than that is lethal over time. The animal that is trivially easy to keep alive in an ocean at minus 1.9 is nearly impossible to ship anywhere. Then there's the ice. Large areas of the Antarctic continental shelf lie beneath floating ice shelves hundreds of meters thick, which means the seafloor underneath them has never been directly observed, and in most cases cannot be. Short of drilling a borehole and lowering an instrument through it. Boreholes have been drilled. Cameras have gone down them. Fish have been seen. But a borehole samples a circle a few meters wide in a system the size of a country. Now recall how the Great Nesting Colony was found. Not by a targeted expedition looking for ice fish nests, by a camera being towed along the seabed on a survey line, in an area where nobody expected anything remarkable, which crossed hours of continuous nests and turned a routine transect into the discovery of the largest fish-breeding aggregation known anywhere on Earth. Sit with the implication. Something on that scale, 60 million animals, tens of thousands of tons of biomass, spread across an area larger than many cities, was invisible until a machine happened to be dragged across it. That is not a story about a lucky expedition. It's a measurement of how thin our coverage is. If a structure that large could remain unknown, the honest position is that we do not know what else is down there. And the fraction of the Antarctic seafloor that has been imaged at all is small enough that the question is not rhetorical. The same limitation applies to almost every open question in this story, and it's worth being explicit about which parts remain unresolved, because the uncertainty is not a weakness in the account. It's the current edge of it. We do not know what a lifetime of accumulated internal ice does to one of these fish. The crystals are real, the resistance to melting is demonstrated, the concentration in the spleen is documented, and the long-term consequence is inferred rather than measured. Testing it properly would mean tracking individuals across decades in an environment where a research season lasts a few weeks. We do not know, with confidence, how the hemoglobin loss became fixed. The deletion is visible in the genome. The mechanism that likely caused it is identifiable in the transposable elements around the break, and the environmental conditions that made survival possible are clear. Whether the loss spread because it was very slightly advantageous in near-freezing water, or simply because a small, isolated population let a near-neutral disaster drift to fixation, is still argued. And the argument is not obviously resolvable from evidence that is millions of years old. We do not know how far the compensations actually go. It's clear that the enlarged heart, expanded blood volume, wide vessels, and supplementary uptake through the skin cover a large part of the deficit under normal conditions. Whether they fully compensate, or whether these animals simply live permanently closer to the edge than their red-blooded relatives, depends on measurements taken under conditions that are difficult to reproduce and easy to disturb. We do not know precisely where the thermal limits sit for most species. Acute values from rapid warming trials exist for a modest number of species. Chronic limits, the temperatures at which an animal can actually live and grow and reproduce over years rather than survive an afternoon, are far harder to measure and have been established for very few. The species that have been tested vary substantially in what they can tolerate and how much they can adjust, which means generalizing from a handful of them to a family of well over 100 species is exactly the kind of extrapolation that gets revised later. And we do not know how the populations will actually respond. That depends on the rate of physical change in specific regions, on how sea ice behaves, on what happens to krill, on whether new predators establish, and on the amount of evolutionary flexibility hidden in populations that have never been tested. What all of that uncertainty does not do is make the underlying picture unstable. And this is where it's worth being careful about how doubt gets used. The specific numbers are provisional. The structure is not. These fish carry deletions that cannot be undone. They occupy a thermal envelope that is measurably narrow. They inhabit the coldest water available on Earth, so there is nowhere further to retreat. They reproduce slowly, and they hold up a food web that has no alternative lineage standing behind them. Every one of those statements rests on evidence that is not seriously contested, which brings the whole thing back around to what this animal actually is. The framing that started this, a fish that breaks the rules of biology, is the version that makes the best thumbnail, and it is exactly wrong. Nothing here broke any rules. Oxygen still had to reach the tissues, and it does, at enormous cost, through an oversized heart and an inflated circulation. Water still freezes at the temperature it freezes at, and the fish did not change that either. It found a protein that could hold a crystal still. Density still determines whether a body sinks, and the animals that rose into the water column paid for it in bone. Every apparent violation, on inspection, turns out to be an ordinary physical constraint being satisfied by an unusual route. And every one of those routes came with a bill that the animal is still paying. What actually happened is stranger than rule breaking, and it goes the other way entirely. This is not the story of a fish that acquired remarkable equipment. It is the story of a fish that lost it. Read the genome as a document, and it is a record of subtraction. A globin gene cluster deleted outright. Its neighbor reduced to an unreadable fragment. Myoglobin knocked out independently in multiple lineages. A heat shock response demoted to permanent low-level maintenance. A swim bladder abandoned before the story even began. A skeleton left half-finished. The one thing that was clearly gained, the antifreeze, was not invented from nothing either. It was assembled out of the remains of a digestive enzyme gene by throwing away the middle and copying a 9-letter fragment until the repetition did something useful. That is a body written mostly in deletions, and deletions only survive in an environment that stops charging for them. Every one of those losses is an accurate record of what the Southern Ocean permitted. Enough dissolved oxygen to make a pigment optional, enough cold to make demand low, enough stability to make an emergency response unnecessary, enough isolation to remove the competitors and predators that would have exploited every weakness within a generation. So the fish is not the impossible thing in this story. The ocean is. An ocean that has held itself within a 2-degree band for millions of years is a far less believable object than a fish with clear blood. Temperature on this planet does not usually behave that way. Every other sea on Earth swings with the seasons, with currents, with storms, with decades. This one sat still, hemmed in by a ring of moving water, cold enough to kill anything without a molecular trick, rich enough in oxygen to make the most important protein in vertebrate biology optional, and closed enough to keep out everything that would have taken advantage. Give an evolutionary lineage those conditions and hold them steady for long enough, and what you get is not a tougher animal. You get a more specialized one. Selection does not build armor against a threat that never arrives. It quietly stops paying for the parts that are not being used, and what is left is a body perfectly fitted to a single set of numbers and helpless outside them. The ice fish is what that process looks like when it runs to completion in a vertebrate, which is why the last thing to understand about this animal has nothing to do with its blood. The remarkable thing is not that it survives an extreme environment. It's that the environment stopped being extreme. From its point of view, a very long time ago, minus 1.9 degrees is not a hardship for a crocodile ice fish. It is the only condition in which its enzymes fold correctly. Its antifreeze stays matched to the water, its heart can move enough thin blood to keep its muscles supplied, and its eggs can develop on schedule. Warm that water by a few degrees, and you have not made life easier for it. You have moved it outside the only set of conditions its body was ever built to meet. That is the reversal the whole story has been walking toward. The fish is not fighting the cold. The cold is the last thing holding it up. So go back down one more time. The lights of a submersible reach out maybe 10 or 20 meters into water that has no other light in it. Beyond that circle, nothing. Above, hundreds of meters of black water, and then a ceiling of ice, thick enough that no sun has reached this seabed since long before anyone thought to look. Small pale flecks drift through the beam and out of it. Ice crystals in water that is already at its freezing point, moving with a current that has been running the same way for millions of years. The beam catches the edge of a shallow bowl in the sediment, about a meter across, its rim ringed with small stones. In the middle of it, a clutch of pale eggs, and lying over them, not moving, a fish with a broad, flattened head and large, dark eyes, gills working slowly, its whole body the color of something that has never needed pigment. It does not flee. It cannot. Half a year of its life is invested in the ground underneath it. Move the light a few meters, and there is another bowl, another fish, another clutch. Move it again, and the same thing. The pattern runs out past the edge of the beam, in every direction, further than the vehicle can travel in a day. An entire city of them holding station in the dark, each one carrying ice in its tissues that will never fully melt, each one running an oversized heart to push clear blood through a body that gave up the one protein everything else on Earth relies on. They have been doing this, in some form, since before there were humans to be surprised by it. And the only reason it works is that the water above them has not moved outside a couple of degrees for longer than our entire lineage has existed. That is the part worth sitting with. Not that the fish is strange, but that it is precise, and that the precision was only ever affordable because something else stayed still. The current is still turning. The ice is still there. The water on that seafloor is still at the temperature it has been at for millions of years. None of that is guaranteed to remain true. And the beam of light that found those nests, the one that put 60 million animals on a screen for the first time, illuminates a circle a few meters wide on a seafloor the size of a continent. Everything in this story was assembled from glimpses like that. Whatever else is down there, holding on inside the same narrow band, has not been seen yet. The dark closes back over the nests as the vehicle moves on. The crystals keep drifting through water that will not let them grow. And somewhere out past the edge of the light, 60 million fish that should not exist go on doing the only thing their bodies have ever been able to do: